Hello everyone! You might be surprised if I tell you that there is a team of students that's currently working on synthesizing an ultimate oxygen booster bacterium. And what if I say that if this so-called super bacteria are not only able to boost oxygen level but are also edible, meaning that they can be consumed as snacks or foods for humans. Sounds pretty insane, right? Then, if you think about it, this blessing of an innovation could be humanity's answer to the ongoing problems of oxygen level, meaning that oxygen level in this world is plummeting, but also it can extend our longevity as a species in Earth, thereby creating a better chance for nature and us, humans. And that's why we are here. CyanoPetals is a group of 9 to 12th grade students that's actively collaborating into making and synthesizing this ultimate cyanobacteria.
Urban planning is a critical aspect of modern civilization, shaping our cities and also communities. However, it comes with a cost that affects the very air we breathe. Currently, urban areas consume about 39.3% of terrestrial oxygen, mostly due to the fossil fuel combustion. This concerning statistic emphasizes the urgent requirement for our awareness of the environmental effects. Moving on to our third slide, we will examine the dynamic relationship between oxygen levels and carbon dioxide concentration. It's crucial to note that carbon dioxide concentration consistently rises as we witness a decrease in oxygen. This correlation highlights the potential consequences of the imbalances in their levels. As we shift to the report given, our focus lands on Jakarta's Air Quality Index as one of the worst in the world. It virtually is a serious situation that demands our attention. But it's not the only problem. We will also discuss about the inefficiency of food production. Managing food resources in underdeveloped regions can be a complex task, further exacerbated by practices that contribute to carbon dioxide emissions. It's imperative to recognize the unsustainable nature of our current food production systems and also explore about the avenues for the improvement.
Cyanobacteria, also known as blue-green algae, are microscopic organisms commonly found in water environment, both freshwater and marine. Despite being referred to as algae, cyanobacteria are actually photosynthetic bacteria, meaning they can generate energy from sunlight through the process of photosynthesis. A distinctive characteristic of cyanobacteria is their ability to produce blue-green pigments that color the water in which they reside. Cyanobacteria have a unique ability to utilize nitrogen and phosphorus in water. Nitrogen and phosphorus are essential nutrients required for the growth of plants and other organisms. However, when an excess of nitrogen and phosphorus is present in the water environment, it can create conditions conducive to excessive growth or blooming of cyanobacteria. This phenomenon is known as cyanobacterial bloom. This blooming can lead to water pollution and various environmental issues. Furthermore, the species of cyanobacteria that we are using can produce toxins that can be harmful to other organisms, including humans. During a bloom, the amount of toxins produced by this cyanobacteria also increases, creating conditions referred to as toxic bloom or harmful algal bloom.
Talking about cyanobacteria produce biomass called spirulina, and spirulina has an amazing potential benefits that we can utilize to solve what problems that we mentioned before. Regarding sustainability, these benefits include: the first one is to produce oxygen, the second one is to be a nutritious meal, and also the third one is to be an appealing food. So let's start from the start, shall we? 200 liters of spirulina can produce oxygen matching with the output of 20 years old tree with minimal energy and effort. This means that spirulina can be a game changer for the oxygen producer. But of course, spirulina isn't here to replace our majestic forest. It's here as a companion. it can add contribution to the important task of oxygen production. And spirulina benefits doesn't stop here. It also offers as a nutritious meal that can be edible, both for livestock and also for human. It loaded with essential vitamins and also essential minerals like calcium and magnesium. And also, not to mention, to progress and cultivate spirulina is super easy and also eco-friendly. Unlike conventional food industry, it doesn't contribute to the carbon dioxide level. And also, the process is fairly easy. We can just cultivate it and also agitate it with essential chemicals, and we will wash it and dry it up, and then we got a spirulina powder that we can eat. And also for the last benefit, spirulina cultivation can be integrated and become an appealing furniture design, paving the way for sustainable home designs and also home products. Not only it can add aesthetic appeals, it can also become an air freshener or an oxygen producer in the corner of your room, or maybe to become a liquid tree in streets. So while the concept might be speculative, this potential can become a life changer for our future to become more sustainable and also eco-friendly.
We are using Actospira platensis to extract its aldolase gene and utilizing Gossypium hirsutum for its NPT2 gene. The restriction sites that are used to retrieve the aldolase gene are INCA1 and BSD API, while the restriction sites that are used to retrieve the NPT2 gene are SPH1 and APO1. Then, these genes will each be gathered in a microtube that contains the required restriction site. Following this initial isolation, a legacy centrifugation process will be implemented to segregate the genetic material based on its size. What we meant by that is the larger DNA goes down towards the palate, while the smaller DNA goes up to the supernatant. Subsequently, these isolated genetic components, aldolase and NPT2, will be combined in a microtube containing the ligase enzyme. This enzyme will bind the two DNAs, a method known as the sticky end method.
Going back to the chases, our team were going to use the plasmid that is originally derived from the Artospira platensis called PJMH020. The plasmid will undergo modification by incorporating the NPT2 and aldolase gene. The NPT2 gene confer antibiotic resistance, while the aldolase gene is going to increase the regeneration speed of RUBP in Calvin cycle. The genetic modification involved the integration of NPT2 and aldolase gene into the PJMH020 plasmid. And to achieve this, we are employing the restriction enzyme called NCO1 and SPH1 to separate the specific site inside the plasmid. This enzyme will create openings that allow the subsequent integration of the aldolase NPT2 gene. The modified plasmid will serve as a carrier for the desired genetic material. The isolated plasmid will be treated with the restriction enzyme called NCO1 and SPH1 inside the microtube. This enzyme will cleave the plasmid at specific site, creating sticky ends that facilitate the subsequent binding of the aldolase NPT2 gene. As you can see, the restriction site is already being replaced by our targeted gene. The modified plasmid will be inserted into the Artospira cell using the electroporation techniques. This involves briefly increasing the temperature to open the cell pores, facilitating the introduction of the modified plasmid. Subsequently, the temperature will be lowered to allow the cell pores to close, trapping the modified plasmid within the cell. Finally, we can see our Artospira platensis, which is being modified and having a feature by overexpressing the aldolase inside of it.
The reason why we are using aldolase is because aldolase is a protein that helps break down certain sugars to produce energy. Glucose is typically generated through reactions and further transformation involving several metabolic steps outside the Calvin cycle. However, aldolase and other enzymes play a role in forming the compounds necessary for glucose synthesis in this process. The conversion of G3P to glucose involves several chemical reactions, and aldolase can participate in one of those steps. G3P, which is the product of the reduction stage of the Calvin cycle, has 3 carbon atoms. In order to form glucose with 6 carbon atoms, several molecules of G3P needs to be combined. An example of step involving aldolase in the conversion of G3P to glucose may include the merging of two G3P molecules through an aldol reaction. This process produces a compound with 6 carbon atoms, which is the basic structure of the glucose. Subsequently, through a series of additional steps, this compound can be transformed into the complete glucose molecule.
Next, NPT2 gene. The NPT2 gene is used as a selectable marker. The primary role of the NPT2 gene in molecular biology is to provide resistance to specific antibiotics, such as G418, by allowing cells to survive and proliferate in the presence of these antibiotics. So here's the illustration. In the liquid of G418 antibiotics, cells that have successfully integrated the NPT2 gene into their genetic makeup gain resistance to G418 antibiotic. In the presence of this antibiotic, these modified cells can survive and continue to proliferate. Conversely, cells lacking the NPT2 gene remain susceptible to G418 effect. When exposed to the antibiotic, those non-modified cells cannot resist its impact, leading them to die or fail to proliferate. The selective advantage provided by the NPT2 gene enables us to easily identify and isolate cells or organisms that have successfully undergone genetic modification.
Okay, so next I will explain about the synthetic biology principles. Our team has designed a benchmark genetic engineering of Arthrospira platensis to overexpress the aldolase gene in the plasmid. The aldolase coding sequence will be fused with the NPT2 selectable marker, yielding an aldolase NPT2 transgene plasmid. This plasmid will be flanked by the NOS promoter and NOS terminator and inserted between the SPH1 and NCO1 restriction sites of the PGMH020 plasmid using standard molecular cloning procedures. Successful integration will place the transgenes under control of the NOS promoter, while the NOS terminator provides an mRNA stabilization function. We will also include a chloroplast transit peptide, which derived from the C. vulgaris RBCS gene to direct the aldolase NPT2 fusion protein into the chloroplast. Overexpression of aldolase is expected to accelerate the regeneration of RUBP, resulting in enhanced photosynthetic CO2 fixation. Meanwhile, NPT2 will allow for selection of successful transformants using the antibiotic G418. This genetic engineering strategy is designed to increase the efficiency of Calvin cycle in Arthrospira, ultimately boosting biomass and biofuel production. Next, I want to talk more detail for each one of the bioparts. The first one is NOS promoter, which derived from the nopalin synthase or NOS gene of Agrobacterium tumaviensis, commonly used for constitutive promoter to drive gene expression in transgenic plants and microalgae. This promoter will also provide consistent, moderate level of transcription in both plant and algae nuclear genome. The second one is the NOS terminator. This terminator is a regulatory DNA sequence derived from the NOS gene of A. tumaviensis, signals termination of transcription and will result in release of mRNA transcript. This terminator also commonly placed downstream of the gene of interest to stabilize the mRNA. The third one is CTP or chloroplast transit peptide, a targeting sequence that directs transport of proteins from the cytosol into the chloroplast. This gene is derived from the chloroplast localized rubisco small subunit or RBCS gene from C. vulgaris. The CTP will also direct the heterologous aldolase proteins into the chloroplast of transgenic Arthrospira. The fourth one is the aldolase, or commonly known as fructose 1,6-biphosphate aldolase. This enzyme will catalyze key reversible reactions in Calvin cycle, and this gene is derived from Arthrospira, and we overexpress it to increase the activity in the chloroplast. The accelerates regeneration of CO2 and acceptor of RUBP will be stimulated after the overexpression of this gene. The next one is NPT2, or neomycin phosphotransferase 2, enzyme that converts resistance to aminoglycoside antibiotics like neomycin and G418. This gene also allows for selection of algae transformants by conferring resistance to antibiotic G418. This will also allow us to use a selectable marker to identify cells that have successfully integrated with the transform gene. And the last one is 35S promoter. This is one of the most frequently employed constitutive promoters for driving transgene expression in transgenic plants and microalgae. And this promoter will also enable successful overexpression of the aldolase gene, which targeted the chloroplast in Arthrospira platensis. So each one of these bioparts can be accessed in the iGEM parts and assembly.